Fine. So we have been talking about different techniques in microbiology research and analysis. So analytical tools we have been talking. So details of electrophoresis, its mechanisms we have discussed, and in the end of the chapter you have advancements in the basic electrophoresis methodology. So wherein you have talked about blotting techniques and hybridization techniques. So further advancement is FISH fish and microarrays, right? So just to begin with the technique in details, just an overview about the subject. To connect with, you can talk about human genome sequence, right? As we all know, it was completed in 2001, and many experiments and researchers have started identifying the regions of DNA and from different organisms, from human genome also, and are they related with any disease conditions, right? And this paved the way to functional genomics, you can say. So what is the study of gene function? So for studying that, parallelly measurement of expression of genome, not only the sequencing but its expression and its functionality, all these things were started getting into the focus of research, right? So how it was done? The most common tools that were used to carry out this kind of measurements like gene functionality, expression measurement of genomes, etc. These are the microarrays, right? So DNA microarrays specifically. When you say DNA microarrays, these can be also called as oligonucleotide microarrays, or they are also called as SAGE, SAGE, that is serial analysis of gene expression tools, right? So when we talk about microarrays, the analysis can be uh, done with the help of DNA. Of course, it is not just limited to find out what is the function of a particular gene, but it is useful in prevention, diagnosis, treatment of various diseases. So not only DNA, but yes, you can take help of various other things which are involved in disease development or normal functionality of the body, right? So it could be a protein also. So when it comes to microarrays, it is not only about DNA microarrays alone, but it could be protein also. So to begin with the discussion, I would like to share the first slide, which is the research paper, right? So by research paper by Sheen et al, and it is talking about glycan microarray story from construction to application. Now, when I say glycan mediated binding process, so methodology, as we will get to know, but why is this word glycan microarray is coming into picture? Because the last word here, glycan protein, right? So a specific type of a protein, and the proteins which are associated in certain interactions, biomolecular interactions. So that also can be analyzed using microarray techniques. So it is not only about DNA, it can be mRNA, it can be protein, so on and so forth. So you can take help of this high throughput method, you can say, which is of importance, and to study in genomics, proteomics, and that is going to give you an overview about the gene functions, their expressions, their association with disease or normal health. So here comes the microarray technology. As you can see here in the slide, it's a glass slide, right? Also, it can be a silicon vapor or a plastic sheet, a fabricated sheet, of course. High quality ones, specialized sheets which are there on the market available. And here you can see small dots. And if I magnify some zone of this plate, here you are seeing each dot is having something like comb. Actually, it is nothing but the DNA sequence, and each component in the sequence, you can see the colored uh, lines are there. One is representing A, T, G, C, like that, the sequences, right? So just as to say ki there are so many oligonucleotide sequences present. That is what is indicated. Now the point is, when we say microarray technology, as I said earlier, it is high throughput method. And though here in the diagram we are seeing a few, two or three in on a dot, but entirely when you talk about the technology, you can um, have an array of thousands or millions of nucleic acid fragments which are attached or immobilized onto the this glass or plastic surface or the silicon vapor. Now this glass, plastic or silicon vapor, what we are talking is referred to as chip, right? CHIP. It is similar to what you have in my computers, right? Chips are there in computers also, and lot of data can be accommodated in those chips. Similarly, here, millions of DNA sequences or fragments can be assembled onto it and analyzed using this. So the chip is what you can have is with the help of nucleic acid fragments, or it can be a synthesized nucleic acid fragment, etc. We will see how the progress has happened in the technique, but yes, to say these are the different sized fragments of nucleic acids which are immobilized onto the solid surface, and that is referred to as a chip. Now, complementary base pairing, that is the second point. The first point was ki what it is, and then how the working. So, complementary base pairing between the sample and the chip immobilized fragments. So, is it not something like hybridization, right? So, advanced version of hybridization, you can say. So, fragments produce light through fluorescence, and that is detected. So, you get to know from these two sentences that you have the immobilized sequences which are going to act as probes, and your sample is added to that, and that is going to hybridize. So when you are talking about interaction to detect, yes, you are going to have some labels attached to them, and maybe a fluorescent label, and you are going to detect the fluorescence using specialized machines. As I said earlier, these microarray technologies, they are used for a variety of purposes in research, clinical studies, measuring gene expressions, detecting specific DNA sequences, then single nucleotide polymorphisms, so on and so forth. Right. So this is the just introductory slide. Now let's go to the second slide where you can see just a diagram. Now you can note down the points what you are seeing in the particular diagram, right? So it will tell you about two streams. One is about normal and one is about tumor. So this is what you are doing is comparison, right? So comparison of sequences. So you are going to check the presence of variation or mutation in the genes which are associated with tumor formation, and it is compared against the normal sequence. So RT-PCR you are going to run. Why it is required? As we know, it is to amplify the number of copies of the particular sequences to be done. So it is done by PCR or RT-PCR, and then you are going to use fluorescent labels. So one is seen as in green and another as in red. So depending upon what fluorescent fluorophore or the agent that is going to give the fluorescence is tagged with your nucleic acid fragment. So accordingly, you are going to get the fluorescence. And here you are combining the two in equal amounts and hybridizing the probe to microarray, and then you are going to do the scan, and this is how the scan looks. So on one microchip, if you see, so many dots are there, and each dot is going to have the probe attached to it. So many fragments of different sequences can be scanned at a time. Right, that is another beauty of this particular technique. Now coming to the details, it is a laboratory tool, but it is a high throughput and comparatively a costly tool, which is used in study of thousands of genes and millions of genes or fragments of DNA that are analyzed together. So DNA microarrays are microscope slides that are printed with thousands of tiny spots in defined positions, and each spot is going to contain particular known DNA sequence or a gene. So that is what is like a probe, right? And these slides are referred to as DNA chips or gene chips or biochips are the different names given to it. So when it comes to DNA chip, it is nothing but the DNA molecule is attached to each slide at a particular point. That is the dot, right? And dot plot technique probably you must be remembering. So you can say it is the very basic approach when it was dot plot, and when I say microarray, it is the advanced format of it. So you can add the sequence which is known to you and going to act as a probe, detect a particular gene expression. So when we say gene expression, transcription is a process. So this fragment is referred to as transcriptome, or you can use mRNA transcripts, right? And you can check the particular um, sequence, right? So most obvious advantage when we talk about microarray is based on profiling of gene expression is very large number of genes in a single hybridization experiment can be analyzed. So millions of DNA copies, as we said earlier, and so many dots. So at a time, you can profile the gene expression, and for different genes, thousands of genes can be analyzed. So now here, this is how it would look, right? So as I said earlier, you are going to use it for DNA, you can use it for assay of mRNA expression, and as the word says, from various biological sources. So it can be tissues, cells, serum, plasma, depends whatever molecule you are targeting. So protein could be there, or it could be mRNA which is coming from the tissue or cells, right? So when it comes to type of materials which are explored in microarray technique, you have variety of the things. It is not only DNA or nucleic acids, right? When it comes to newer microarray applications, they include measurement of gene copy number, that is what we said, profiling the combination of SNP, single nucleotide polymorphism. So if there is any variations in a particular sequence, maybe a single base change also can be detected. So polymorphism leads to variations in a particular species. It can be positive, it can be negative. Generally we say it as a mutation, but mutation and polymorphism, SNPs, you have divergence from the single original species, and particular sequence at a particular gene locus is varying, and you get a diversity or variations in the particular expression. So SNPs detecting that is important. So that can be done. Genome structure, composition of different organisms you can check, or cell types also you can match and find out the similarities and variations. Alternative splicing at exon and intron junctions. So n number of applications you have when you talk about microarrays, right? So this is how the chip would look. It is very small, like your admit card size as it appears in this particular picture, right? Now when I say types of microarrays, we were talking about, so of course, again to summarize, it is multiplexed, right? It is not a single analysis. You have multiple possible sites where the analysis can be done. So it is multiplexed lab on a chip. That is how it is described as. So you can call it as microarray, 2D array. Solid substrate is there. How the design is, that we will see. But when it comes to types, as I said earlier, it can be called a gene chip or DNA biochip, DNA microarray. But here, when I'm talking about the types, let's see the point number 4 in this. So DNA microarrays, when I say, it can be simple nucleotides, oligonucleotides, or fragments of a particular gene, or it could be cDNA microarray. When I say cDNA microarray, you know mRNA is converted to its cDNA using reverse transcriptase, and that DNA is used in microarrays as probes, or oligonucleotide microarrays. That is one. Then back microarrays. Have you heard about YAC and back? All right. So back microarrays and SNP, that is single nucleotide polymorphism microarrays. So chips. So when it comes to surveillance of microRNA populations, that they are referred to as chips. So all these are the versions of microarrays which are used in genomics and proteomics. You are going to have protein microarrays, peptide microarrays, which are going to elaborate on analyze protein-protein interactions, right? Tissue microarrays are there, cellular microarrays are there. Initially, antibody microarrays were there. Of course, any chemical compound also can be used, or further as a glycan-based microarray, that lecture we started with. So that could be there, or any specific biomolecule can be used. So glycoarrays that we talked in the beginning, so carbohydrate arrays, phenotype microarrays, reverse phase protein microarrays, so many types of microarrays are there. And of course the probing material and the target molecule that is under analysis, these are going to vary, but basically it is a glass or a silicon vapor or a plastic sheet, right, which is going to act as a solid surface, and you are going to proceed with hybridization or interaction between the molecules that is explored in this, right. So when I say about these uh, microarrays, here is, oh, I'm sorry, just give me a second. Yeah, here is the general flow chart of the steps, right. When I say mRNA, that is gene expression, I want to check. So I'll go with extraction of mRNAs from a particular tissue or a cell, and then you are going to uh, use it for analysis. Now, this mRNA could be obtained from normal tissues, which is going to act as a reference, and from the disease or suspected or patient's tissues, which is going to show certain abnormalities or degree of expression would vary, and so on. So, experimental sample that you are going to call it. So, this is what you are going to extract. Then these two mRNA samples are converted into complementary DNAs. So, mRNA is converted to cDNA. Now, each sample is labeled with a fluorescent probe, because hybridization, if it is going to happen, it is to be detectable. For that, you need a label, and that is a fluorescent probe, which is commonly used, and yes, of different colors that you are using, right? So, in the previous diagram, also, previous slide, also, you have seen one as a green and one as red. So, what kind of chrome you attach to it, accordingly, you will get the difference. So, for example, in the experimental cDNA sample, we label it with red fluorescent dye, and the reference cDNA is labeled with green fluorescent dye. Just imagine this, right? So these two samples are then mixed together and allowed to bind to the microarray slide. Now, the process in which the cDNA molecule binds the DNA probe on the slide is called hybridization. It is similar to what DNA-DNA hybridization or DNA-RNA hybridization we talk in. It is similar to that. So following hybridizations, the microarray is scanned and you are going to measure the expression. So intensity of fluorescence will tell you about the expression levels. Right. Now, how to analyze this? You have put a red labeled sample and reference is in green label. So let's see what kind of expression you would see. If the expression of a particular gene is higher in the experimental sample than in the reference, then the corresponding spot on the microarray will appear red, right? And if it is opposite, that is, if the expression in the experimental sample is lower than in the reference sample, then spot will appear green. And of course, equal expression will give you some another color, which is yellow in the expression. Of course, what fluorochromes you use, based on that, these colors you will get, because we had taken fluorochrome dyes which are green and red in combination. These are the outcomes, right? Now the colored or fluorescent spots on the surface of the microarray, you are going to uh, observe or scan, and that is going to tell you about or indicate about the location of hybridized molecules. So the light signal which is produced by label is analyzed by scanning with the help of photometry, right? So you are going to require a specific scanning instruments depending upon what kind of labels that you have used, right? So it can be photometry if you are using the light generated light measurement. So fluorospectroscopy also, you can say, it can be used for detecting the signals that are generated, right? And these data is gathered and then it is used to profile the gene expression. Of course, this data analysis is done with automized computerized systems. Now here there is one more square that you are seeing. Capture probes immobilized on the solid substrate are exposed to the labeled sample under conditions. It is very important that you are playing with biomolecules, so interactions are to be conducted at controlled conditions. So what are the specific conditions which are important? So specific buffer conditions, temperature, linkers, all these things would be required to allow the hybridization of these two complementary molecules. So this is uh, very important that under what environmental parameters you are going to execute the hybridization process. So in short, to say what you have a sample molecule that you have extracted, that you have purified, you have amplified, and you have added it onto micro chip. Hybridization is allowed to occur. Then of course washing is going to be intermediate step to remove the unwanted or unused unhybridized molecules. So washing is done, and then you are going to take it for scanning. So this is the overall flow chart for the process, and it can also be represented in this way, right? So why I'm saying this way, it is here you are doing comparison. Now in this case you had only one of the sample and another was reference, so this is one channel system, है ना? But when you can have two channels or multiple channels, how the process would look like. So here is the schematic representation that shows here are multiple rows, right? So multiple channels are there. What are the channels? The channels are indicating nothing but you have extracted the sample from different sources. So RNA sample 1, RNA sample 2, of course not necessarily it is going to get restricted to 2. You can have it as many as possible. All right. So here I'm talking about the case where I'm going to have two different samples. So isolate RNA, assess the purity and integrity. That is very required. So at every step, you have to have very stringent analysis with respect to the particular step. Now, using this RNA, what you are going to do? You are going to produce cDNA synthesis. You are going to do reverse transcriptase process. So here, cDNA sample 1, cDNA sample 2, you will get respective to the RNA samples that you have obtained from the different sources which are be compared. Right. Now here, because the samples are differing, you have to use different labels. Now, CY5 labeled cDNA or CY3 labeled cDNA. What is this CY5 and 3? That is one thing which we will talk when it comes to different probes and materials. But let me tell you, not only fluorescent dyes, but the targets also are specific. Generally, cysteine is utilized. That is going to be tagged. So accordingly, you will find variations in these numbers and the titles which are given here. So here Psi 5 and Psi 3, these are the labels used, but which label to use, where to tag, all these details I'm not going to talk right now. But yes, this variation is to be taken into consideration. Now because here you have two different source samples, it is two channel system we are talking about, and then you are allowing the hybridization to go. You are applying these two samples which are differently labeled with different fluorophores to the microarray, and differential hybridization will occur, and it would give you differently colored spots, maybe color or fluorescent, depends, right? And then you are going to go for image analysis using software. So in the process, if you see that a sequence on an array hybridizes with one, or it may hybridize with both, right? It is possible, or it is quite possible that it will not hybridize with any of the probes. So if you are using CY5 label, you will see red color spots. If you are using CY3, you will get green color spots. Both the probes together, so it is binding to both, you will get yellow color. And if it is not binding to any of the probes, you will get no color light produced over there. Now these are the things which are going to depend on what label you are adding. But here what is the concept? In the previous case it was single channel. In this case it is double channel or two channel system or dual channel system. Like that you can have multi-channel systems also. Now when it comes to microarrays, I'll come to this slide little later. Let me talk about this. Right now what you are seeing in this diagram is, this is how the DNA microarray technology schematically represented. So again, the very first slide that we had started with. So this is the normal, this is from the tissue or sample taken from the patient, and these are the microarray chips, right? These are the machines, how they would look like, and what you get is this scan, and the scan is analyzed. So when you talk about the uses, First, they were introduced for DNA, right? So as a search tool, but with mm, advancement in the technique, large-scale population studies also can be done with the help of microarray techniques. For example, to determine how often individuals with a particular mutation actually develop breast cancer. This is one of the examples. So single nucleotide polymorphism can lead to variations, and variations could be good, or that will generate, say, the patterns in the insects, shell patterns in the certain aquatic animals, or it could be leading to certain disease conditions. So single nucleotide variation can lead to development of cancer. BAR genes are there, and single nucleotide variation can give the disease development or cancer development. So a large population can be scanned for that BAR gene, and single nucleotide variations in the particular mutation site can be checked uh, for the for the possibility of developing the breast cancer. So not only breast cancer, but yes, you can similarly you can see the changes in the gene sequences that are associated with any particular disease. Right? Microarrays can also be used to study the extent to which certain genes are turned on or turned off. That is what we talk with reference to mRNA produced. So gene expression cases. So here, instead of isolating DNA, we can isolate RNA, and then after isolation, you can measure the volume of mRNA and the amount of cDNA. Of course, it is not truly a quantification technique, but yes, it can be used to check the extent of gene expression. DNA microarrays are used in clinical diagnostic tests for diseases. That is one thing we talked. Results from microarrays help to understand complex biological systems in drug discovery, for example, for elucidating the molecular basis of diseases, interaction of drug with the target molecule. All these things can be, because it is interaction between the two complementary molecules, right? So hybridization process not necessarily has to be there between the two nucleic acid samples, one as a probe and complementary sample DNA, but it can be between any molecule, and which is having a complementarity for another neuron interaction for the another one. That's why we have antibody microarrays also, or protein microarrays also. So it also can be used to determine the molecular basis of the disease. It can be used to determine the drugs which might be prescribed for a particular individual, because genes determine how your bodies handle the chemistry related to those drugs. So in drug discovery, drug application, uh, drug sensitivity in individual cases, right, that also can be analyzed using microarray techniques. So when it comes to microarray as a biosensing technology, let's watch a video if yes. Sorry. Yeah. This is how different color spots will develop after the reaction. So here we are taking example of budding yeast cells, right? Gene expression is allowed, activated, or repressed. These are the two conditions. So here are the ependroptives having the cells which are growing aerobically or anaerobically. Right? Yeast cells you are seeing in that. So you are going to isolate the mRNA from these cells. Right? extracted the mRNA from the cells. You can see the color changes that indicates the process is going on. We are extracting the mRNA. Of course, these are indicative steps. It's an animation. Now these are the mRNAs, right? From each tube, you are going to have many mRNAs like this. Yeah, they are converted into cDNA now, right? 6,000 dots are there on this particular slide. And out of these, which are giving you what kind of a color. So each of the spot is made up of DNA and that can interact with your sample. Now this sample is loaded. The chip is already ready, right? DNA already is there bound on it. This is your sample molecule which is coming and binding with it. And because of the fluorophore that is attached to it, you will get different colors like red here in this case, if it is complementary. Green in this case, if it is complementary to this. So sample one, sample two, like that, since you are going to get. So now this is the analyzer or scanner, and that will tell you about bound cDNA. To visualize it, scan it, these kind of machines are there, right? It is going to scan it, and this is how you are going to see it on the screen. What does it tell you? Both are green, right? We had seen, okay, now this is about red. So you are comparing normal and reference or diseased and reference like that. So this analysis can be done. These are the basic steps to visualize. Yeah. Here if it is there is no interaction or equal expression, you get yellow color. Less expression, you get red. High expression, you get green color. So only in aerobic cells, only in anaerobic cells, and expressed in both. So type of mRNA that was under study is checked for its expression in aerobic cases or anaerobically grown cells. Okay, so here it's the overview of the technique, right? So as you must have got to know, these are the biosensing technology, that is what we say, and you have a solid substrate like plastic or glass slide covered by molecular capture probes. The molecular capture probes may be DNA, RNA, oligonucleotide stretches, right? A protein, antibodies, or glycan-like molecules. We have seen the types in the previous discussions. So small amount of sample is exposed to array under correct conditions for hybridization, and interaction between the capture probe and the analyte is observed using the scanning instrument. It is as simple as that. Right. So when it comes to making performing this analysis, what are the steps? Sample preparation. So as we have seen, extraction of that RNA and its purification and its check for integrity. That is sample preparation. Right. Then you are going to have second part in sample preparation, that is labeling step, right? And then you are going to add it to the chip. Hybridization is going to be allowed. Then you are going to wash. So washing steps are there to remove the unwanted, unattached, unhybridized molecules. Then you are going to go for optical scanning, which may be based on absorption or maybe based on fluorescence, depending upon what kind of label you have used. And then data analysis. So as I said earlier, the capture probes could be biological molecules like PCR products, nucleic acid sequences, DNA, RNA aptamers, oligonucleotides, proteins, antibodies, small molecules, or carbohydrates, depending on the goal of your analysis. So this is one summary slide, you can say, which is telling you about everything. Now this is what you are going to schematically represent the biochip. So you are going to have so many spots on one slide, and they are numbered as A1, B1, C1, D1, like that. So this is row 1, row 2, row 3, like that. So on the surface of a microarray chip, there are large number of spots, and each are going to contain several copies of each single capture molecule that we know. And spots are coded to allow identification. So each spot is specified by its coordinates in the 2D array. Now, for instance, in a particular array, it is clear that the spot with the coordinates x1, y1. So if I take this first one, so this is y and this is x, as in the graph we say ki horizontal axis and vertical axis. So x1, y1 accordingly that. So both are 1, so it is a1, right? So like that you can have the code for all of these, and then when analysis is done, you can correlate with the sample that was used. So this is how you can schematically show while writing the answer.